In the field of ecology, one of the most important things is the collection of data regarding populations. In order to study a particular habitat, the populations of species must be counted. However, due to constraints of time and effort, not every single organism can be counted. Therefore, a method of sampling must be used in order to obtain a representation and estimate of the number of species that exist. There are two main methods for sampling that are commonly used by ecologists, the first being random sampling. In order to carry out random sampling, a quadrat must be used. A quadrat is a square of a set area that is used to isolate the area in which the measurements of an individual sample are taken. In order to take samples using a quadrat, first the area over which samples will be taken must be marked out using a grid system. A random number generator can then be used to calculate random coordinates, and samples are taken using the quadrat only at those coordinates. In order to gain a representative sample size, a few factors must be taken into account. Firstly, the size of the quadrat. This is chosen based on the size of the organisms predicted to be sampled. Larger organisms will need larger quadrats, but the spacing of organisms also plays a part in deciding the size of quadrats. For organisms that are spaced in small clusters, a large number of samples taken with smaller area quadrats will be more valid than a small number of samples taken with a quadrat of a larger area, even though the total area sampled may be the same. A large enough number of samples need to be taken using the quadrats in order for the data collected to be reliable, and when taking samples with the quadrats, each section within the given area being studied should have an equal chance of being selected to be sampled. A second method of sampling is also appropriate for different regions, particularly when there may be a graduation across the area in the number or type of species present, for example in a sand dune environment near the coast. This method of sampling is known as systematic sampling, and it involves creating a transect of the area. This can be done as a line or belt transect and involves laying down a section of tape of string and counting any organisms that fall within the belted section or along the line. This is a convenient way to show a graduation or trend in species or abundancy, as different areas may have different densities. Quadrats are typically only able to be used for non-mobile organisms such as plants, because mobile species may move out of the sample area, which makes it more difficult to ensure a valid result from the sampling method, as organisms may be counted more than once. Between deciding whether random or systematic sampling is more appropriate for a region, several factors can be used to decide. Random sampling will be more appropriate when there is a relatively even distribution of species throughout the entire area over which samples are being taken. Systematic sampling, on the other hand, is better for when a gradient of species is present across the area to be sampled, because this method allows that transition to be captured in the data. Within systematic sampling methods, using a belt transect rather than a line transect will provide better information on density of species within the sample space, as it collects data from a greater area. However, as these sampling techniques are not ideal for animals, only plants, another method must be used in order to measure animal populations. This is the mark-and-release technique. This process involves taking one sample of a population in an area and then marking those captured and recording the amount captured. After a short period of time, then a second sample is taken under the same conditions. This time, the total amount captured is recorded, and the number of those that are marked can be used to estimate the size of the entire population in an area. From the second sample, the total amount captured is divided by the number marked, and this gives us an estimate for how many organisms of a species there are. For every organism that was marked, this is then multiplied by the total captured from the first sample to provide an estimate for the population. Larger animals such as bears or wolves can be marked with a collar, or smaller ones such as snails or insects can be marked with pen or stickers. Locations can be chosen randomly or systematically, similar to quadrats within an area. The benefit of this method is that the researcher would have very little impact on the results from their own bias, as it is largely reliant on chance. However, it can still be affected by the researcher, like if they choose a location with an unusually dense population, or if they scare animals off. These would all affect the total estimate. Care has to be taken on how the creatures are marked as well. It could make them more visible to predators, or could even be toxic. Once data has been collected by either of the two sampling methods, it must be analyzed in order to assess its validity and reliability. There are several statistical tests that are used to do this. The first common analysis is to find the standard deviation of the results. This involves calculating the mean of all the data collected. In the example shown, this is shown to be 6.3, and it is found by adding the total number of species found from all the samples taken and dividing by the number of samples taken. Each individual value for each sample is then taken and the mean is subtracted from that, then this result is squared. The total of the squared values is then added up to include the data from every sample taken. This number is then divided by the total number of species counted minus 1 to find the variance, and the root of the variance is found as the standard deviation. Within a population, a bell curve will represent the data collected. Within one standard deviation of the mean, 64% of the data will fall. Therefore, in terms in random sampling, this means 64% of the quadrats will contain plus or minus 1 standard deviation value from the mean number of species within the quadrat's area. Within 2 standard deviations, 95% of the quadrats contain plus or minus 2 standard deviation values away from the mean of the number of species. Standard deviation can be used to assess reliability because a smaller standard deviation value means there is less deviation from the mean. Therefore, this means across all the samples taken with a quadrat, they all yield a similar number of each species within the area. This equates to an assumption that the distribution of that species is fairly even and means when an estimate for the total number of that species within the entire area is calculated from the mean is more likely to be accurate. A second statistical test that can be used is known as the t-test. The t-test is used to compare two different groups of data. For instance, data collected in the same way from two separate locations or at two different time periods, or data collected from the same area using two different methods, such as random sampling and systematic sampling. The test determines whether the data from both groups are statistically different to one another. The test takes into account aspects of how much variation exists between the two means of each group and how much variation there is within each group itself. The value obtained is a ratio between these two factors. Once a value is calculated, it can be compared on a table of significance in order to determine if the difference between the two group S values is large enough to be significant. This means the difference is large enough that it could not have been caused by chance, and therefore suggests the presence of another factor that is causing the variation. Data obtained from random and systematic sampling can be used to calculate the abundancy of species within an area. This is the total number of individual members of a species that is found in the area, and it can be measured in several ways. The first method of measuring abundancy is by using frequency. This is a measure of how often a particular species appears in a sample. If the sample appears in 1 out of every 2 quadrat samples taken, then its abundance frequency is 50%. This method, however, does not give much detailed information on the density of species within the region. The second method of measuring abundancy is estimating the percentage cover. This is useful for if a species is hard to count or particularly abundant. This allows for a much more rapid collection of data, as each individual organism does not need to be counted. Thank you for watching.